Okay. Hello, everyone. I am Dr. Rahul Suryawanshi, working as assistant professor at College of Veterinary and Animal Sciences, Udgir, teaching the subject of veterinary public health and epidemiology. Today, I'm going to teach you guys about this particular subject on, in particular, a food safety aspect. I have broadly divided this subject as a veterinary public health and epidemiology in 4 parts. Let's see. As you can see on this particular slide, teach it, and the reflection you can see it's learn. You can see teach learn means it's a reciprocating thing. If someone is teaching, and unless and until you are absorbing the thing, you are in a learning mode, it's not possible to teach anything to anyone, right? So let's start with the thing. So as I said, I have divided this subject public health, veterinary public health, in 4 aspects. That is, first thing will be on veterinary public health and food safety. Second thing will be the unit 2, like veterinary epidemiology. Third one will be on zoonotic diseases. And fourth one will be on environmental hygiene, which is a unit 4 in as per our syllabus, right? So let us start with the first part, that is the food safety thing. As you know, the veterinary public health is a multidisciplinary subject, and as a multidisciplinary subject, it touches all these aspects, and all these, whatever syllabus has been assigned in veterinary public health will be touching to all these topics. You can see it will be like food hygiene and food safety, zoonosis, their control, epidemiology, disease diagnosis, biomedical research, environmental hygiene, collaborative activities, and the most important thing in a country like us, like, you know, it's a developing country, public education and training is one of the key component which is assigned to this particular subject or a public health veterinarian. Without wasting a time, let us start with today's subject. That first thing we will be talking about food safety. The foods which are assigned to our syllabus will be mainly like a milk, meat, egg, and fish, right? So let us start with the milk hygiene. What is milk hygiene? It is an expert supervision of all milk and milk products with the object of producing wholesome milk for human consumption, preventing danger to the public health. Means the journey of food. It might be milk, it might be meat. The journey of food right from the production till it reaches to the fork, means farm to fork, or till it reaches to the consumer in a safe and hygienic manner. Whatever things as a quality control officer or a quality being a veterinarian, looking after the quality control aspect of this particular thing in a processing plant, we are supposed to be very vigilant about this thing, to take care of all aspects and the critical control points of the journey of these raw products till it becomes the product, right? Then we will be talking about the zoonosis. These are nowadays everybody is very much quite acquainted with this particular term. These are the diseases and infections which are naturally transmitted between vertebrate animals and man. Means there are more than 700 diseases which are shared by animals and humans. Then environmental hygiene. It's everybody knows about so many the basics of the environmental hygiene, but in this particular course of veterinary public health, in the environmental hygiene deals with the all those factors like the surrounding or physical environment of human or animal, which may have deleterious effect on human and animal health and well-being. In particular, when we are talking about the industries and the the water coming out, sewage coming out of that particular industry, if it is directly mixing with the natural water bodies, definitely it is causing a deleterious effect. And we are being veterinarian, that aspect of environmental hygiene we are going to learn. Then epidemiology is nothing but the study of distribution and patterns of health related events, health characteristics and their causes, and those are the influences in well-defined population, right? When we are talking about epidemiology, suppose there is some outbreak is going on, and being a veterinary officer, you are appointed to investigate that outbreak. Whatever terms and the basic concept you will require to know, and to write down your report, all those concepts are taught in this particular aspect of public health, that is epidemiology, which includes disease etiology, outbreak investigation, surveillance, screening, biomonitoring, comparative treatment effects such as clinical trials. Means if you are having, if some unknown disease is there and some remedies are there, there are two kind of remedies, and if you want to compare with the outputs of those remedies, then it comes under like a clinical trial. Means you will be making two groups of animals, and those clinical trials of those remedies will be taken, and based on their result you can recommend that which one is better one. Then definitely when we are talking about veterinary public health and its significance, and in particular when we are dealing with the competitive exams, definitely some key things will be of importance, which we will be discussing in today's lecture. You'll realize, before preparing this particular lecture, I talked with my PG students and all those juniors, those who actually appeared for LDO exam, and I took their guidance while preparing this kind of presentation. In particular, the aspects I tried to cover, the brush up the food safety aspect of in toto the veterinary public health. So we will be touching all those aspects which are very crucial or important so far the food safety is concerned, right? So coming to the historical significance, definitely from Vedic eras, Romans, Egyptians, Arabic, those everybody, all those civilization which evolved along with their sciences, in their evolvement, they definitely deal with the concept of the One Health and the public health. The William Shobe was the person in 1940 who gave the concept of one medicine. One medicine is a concept which throws light on the concept of one health. We will discuss that particular thing in second lecture. Then the concept of VPH, it was evolved at CDC in 1940s. I'm going to share this particular presentation with you guys, so that I'm not going to discuss each and every point because it will take so much time. So definitely we will just go through that presentation and the important aspect or the issues we will discuss here, right? So in 52, definition of VPH was given. Kaplan was the person, Mr. M. Kaplan from WHO, who gave the modern perspective. So far, Indian history of VPH is concerned. In 64, NICD, a division of zoonosis, was established. 65, Pantanagar, at Pantanagar, VPH degree was started. In 71, at Kolkata, a master's degree program was initiated. In 71, IVRI, division of VPH was established. Like away. Ahead. Then let's talk about the contribution of others in veterinary science. Although these are the scientists which were not veterinarian, but their contribution has made a revolution in the veterinary science, which includes the Louis Pasteur, who invented the process of heating of wine to retard the fermentation, Mr. Soxlade, who gave the concept of pasteurization of milk, Mr. William Osler, a physician from Johns Hopkins Institute, who significantly contributed in development of VPH and investigation of like diseases like anthrax, tuberculosis, and cholera. And everybody knows very much, everyone is very much familiar with the Koch postulates. Then Smith, who gave the invented the Salmonella species. And very recently in 2016, Chen, Murphy, and Sampolesi, these scientists, they gave the stem cell therapy, which is actually used for various ailments of animals like myocardial infarction, ligament damage, and many more. There are certain veterinarians who actually contributed in human health. Although they are not, although they are veterinarian, but their contribution is playing major role in the having revolution in human health. Like Jacob Neuffer, who was a veterinarian, who in 15th century only, he performed a first successful cesarean operation in woman. Then Edward Jenner and many more, all those Loeffler, Frosch, all these veterinarians. These are the list. You can afterwards you can read out all those things when I will be sharing the PPT with you guys. Let's go ahead. Let's talk about milk. Definitely, we will be starting with milk, meat, stunning, and all those procedures. Okay, milk. So, as per PFA rule 1976, milk is legally defined as a fresh, clear lacteal secretion which is obtained from complete milking of one or more milch animals. That too 15 days before and 5 days after calving, or it should be free from colostrum, and it should contain prescribed amount of fat and SNF. In general, everybody should know that cow is having fat percentage like 3.5 as compared with buffalo, which is 6% in buffalo milk, and 8.5% SNF in cow and 9% SNF in case of buffalo milk. When we will talk about the composition of milk, milk is broadly divided in two parts: water and total solids. Water is again subdivided in free water, bound water, and total solids are divided in SNF and fat. Fats further divided in true fat and associated substance like vitamins like A, D, E, K and phospholipids. These true fats, they are further divided in simple lipids, compound lipid and derived lipids. Simple lipids again further divided in SFA, UFA. Again, SFA is having two types, like a short chain and long chain. Talking about the SNFs, which is very much countable so far when we decide the quality of milk. The SNF, it contains lactose, nitrogenous substances and other substances. Like lactose will be like alpha lactose and beta lactose. Nitrogenous substances will be like protein and non-protein substances. Non-proteins like casein, alpha, beta, gamma, and then VA, serum and some globular protein. Then non-protein part which contains urea and amino acids. Other substances in traces will be like minerals, enzymes, pigments, different vitamins, gases and globulins. When we talk about the constitution of milk, there are some concepts which are very important. So far the competitive exams are concerned. The concept like lactose intolerance. You know, lactose intolerance is nothing but the inability of adult to digest lactose, a sugar found in milk, to a lesser extent dairy product, causing side effects. And this particular lactose intolerance, it is also called as they won't be asking about the lactose intolerance. They will be using the term hypolactasia, right? Like a lactase deficiency or hypolactasia. Again, this lactose intolerance, it is a phenomenon which is observed because of an enzyme which catalyzes and the hydrolysis of lactose, which converts lactose into glucose and galactose. When this particular enzyme is not present in or insufficient in level, then such kind of thing or such kind of phenomenon is observed, right? Again, when we talk about the clinical symptoms because of lactose intolerance, then it contains like abdominal bloating, cramps, flatulences, diarrhea, nausea, and borborygmy, which is also known as rumbling stomach or vomiting after the consumption. Means that particular person will not be able to digest the milk. Again, as an option, he can go for the plant-based milk like soya milk and all that, right? Gases, again, when we talk about the gases in milk, there is one phenomenon called as rectangle phenomenon, which is very important so far these competitive exams are concerned. This rectangle phenomenon is based on the specific gravity. When we say there is a test, when we calculate, we judge the quality of milk based on specific gravity. Specific gravity of freshly drawn milk is 1.030, while the same specific gravity of same milk after some time will be like 1.025. Means it's a stale milk. To identify a stale milk, you can take help of this particular phenomenon of milk called as rectangle phenomenon. Then when there is some rotten egg like smell, definitely there is a production of H2S, and it must be because of the action of microorganism. And another important thing to be kept in mind when someone talking about rectangle phenomenon, specific gravity of colostrum cannot be compared with this particular phenomenon, because specific gravity of colostrum will be like 1.079 and it will be quite different. You can't compare with the normal milk. And even while writing about the milk definition of milk, legal definition of milk, we saw it is not they have intentionally separated this particular thing like colostrum from the milk. Normally it's not a normal milk. This is a particular lacteal secretion for the kid, newly born kid. Or then globulins, definitely globulins, they are in globulins, immunoglobulin A and immunoglobulin G are present in freshly drawn milk, and these A and G immunoglobulins are maximum in case of colostrums. Then in composition, the milk has its own power of it's preserving itself, like its shelf, increasing its shelf life. It's like up to 3 to 5 hours, right? Based on its own inherent things, like those in antimicrobial substances in milk which are naturally occurring, can be classified in two parts, like specific factors produced in response to specific antigens and non-specific or less specific antimicrobial activity, right? This A part is having inherent or naturally occurring substances, and another thing which there are things which contribute in increasing the shelf life of milk, but they are non-inherent, which are not naturally occurring into milk, but yeah, definitely they are contributing, like antibiotics, sanitizers, and other chemicals. Okay, let's talk about these things like specific factors and non-specific factors ahead. So specific factors will be like definitely immunoglobulins, which are responsible for maintaining the immunological protection of neonates through the colostrum to newborn. Definitely it neutralizes toxins and viruses, prevents absorption of food antigens, suppresses bacterial growth. Concentration of immunoglobulin A, M, G will be, when you compare this particular thing with normal milk, will be in a very extensive quantity in case of colostrum. Like immunoglobulin A will be 100 to 700 milligram per decibel, and in M will be 300 to 1300. Immunoglobulin G will be 3400 to 8000. Again, another factor which is present in case of human milk and not present in animal milk, but it is discussed, and many times it is very important so far the competitive exams are concerned. Bifidus factor. Bifidus factor, it is responsible for the dominance of bacterium, Bifidobacterium bifidus in infants, in humans only. It is advantageous for babies. This particular factor, it inhibits the enteropathogenic E. coli, like E. coli, Shigella, and other pathogens because of the acid. The acidic pH, it helps in absorption of calcium, phosphorus, as well as iron. And this bifidus factor is also responsible for the synthesis of vitamin B as well. Then lactanin is there, which is a non-specific factor, active. It is active against Streptococcus pyogenes only, because this is bacteria specific, you know. Then the bovine colostrum, which is rich in L1, and normal milk, which is specifically rich in L2. Recently, fraction L3 is also detected so far this lactanin is concerned. Then factors affecting lactanin activity, again, based on other temperature, like temperature kind of thing, heat treatment, 70 degrees Celsius for 20 minutes, again the pH, all these things. L1 completely destroyed, leaving behind the L2 fractions when you are subjecting that milk to heat treatment like 70 degrees Celsius for 20 minutes. Then components like cysteine, hydrogen peroxide, then p-chloromercury benzoate destroyed the activity of lactanin. Lysozymes, another important thing. Human milk contains 30 milligram of, per 100 ml, which is 3000 times more than the bovine milk. Means the human milk is very much richer in case of lysozyme as compared with bovine milk. It protects against enteric infection, destroys many of the gram-positive bacteria. Then lactoferrins, they are also known as iron-binding proteins. They are also called as lactotransferrin or lactosiderophilin. This lactoferrin is mainly in a whey protein, contains 70% of whey protein and 30% of bound with casein and fat. Human milk, it contains high amount, 1.2 gram per 100 ml as compared to bovine milk, which is 100 mg per 100 ml. The function of lactoferrin is basically to bind with iron, bind with iron and making it unavailable for the bacterial growth. And the organisms which got prohibited because of the lactoferrin will be like Staph aureus, Pseudomonas aeruginosa, Vibrio, Clostridium, Bacillus cereus, and Bacillus stereothermophilus. Then a very important thing, which is also known as lactoperoxidase, which is very known as lactoperoxidase system, LP system. It is present only in case of cow milk, which is 30 mg per ml. But this particular thing is absent in human milk. When we were learning about the bifidus factor was present only in human milk and absent in bovine milk. But this lactoperoxidase system is present in bovine milk, but it is not present in human milk. Again, the inhibitory action is due to formation of antimicrobial system called lactoperoxidase thiocyanate hydrogen peroxide system or LP system. Each and every thing in this system has its different role subscribed. Like the lactoperoxidase will be acting as an enzyme in this chemical reaction, having an iron containing heme group. Then thiocyanate will act as a substrate, which is 1 to 10 milligram per kg. Hydrogen peroxide will be acting as a promoter, which is contributed to polymorphonuclear PMN or H2O2 producing other bacteria. Then the bacteria whose growth get retarded because of this LP system will be streptococci, and the bacteria which gets killed with this LP system will be E. coli and Salmonella typhimurium. Then are certain vitamin binding proteins, they also play major role. Fatty acids, major fatty acid like saturated fatty acid like octanoic and decanoic acids, having some long chain unsaturated fatty acids. Some miscellaneous enzymes are there which also contribute in antimicrobial activity of milk. There are almost 23 enzymes which are identified in milk. Then talking about the non-inherent, as I said that these are not naturally occurring in the milk, but in course of journey of drawing milk from udder of milk animal till it reaches to the consumer, these at certain point of critical control points, these things get added to the milk, although they are not healthy for the consumer, but they definitely contribute in increasing the shelf life of milk, like antibiotics, sanitizers and detergents, pesticides and radionucleotides. Then let's talk about the microflora of milk. Microflora of milk, it contains bacteria, yeast, mold, viruses, and again a very important aspect of viral microflora will be bacteriophages. We'll see in detail ahead. Then this microflora is basically containing the different kind of a heterogeneous population of mesophilic, thermophilic, thermodiuric, psychrophilic, as well as pathogenic microorganisms. Okay, let's see what is the difference between these organisms. Let's see. When we talk about the mesophilic bacteria, these are the organisms which can grow at 30 to 32 degrees Celsius, normal temperature, which actually observed at normal room temperature, what we can talk about. Again, mesophilic bacteria, those mesophilic bacteria can be a gram-positive cocci, those can be asporogenous gram-positive rods, and some spore-forming gram-positive rods. Again, these the sporogenous gram-positive organisms mainly contains like Mycobacterium bovis, Mycobacterium tuberculosis, Corynebacterium diphtheria, pyogens, and bovis. And the spore-forming positive rods, they consist of Bacillus cereus or Bacillus subtilis. Then gram-negative rods are also there, which are mesophilic in nature. Pseudomonas is there, Pseudomonas species, almost Klebsiella and Alkaligen, Acinetobacter. Again, coliforms and fecal coliform. Again, coliforms will be, these are the aerobic facultative anaerobes, but those will be lactose fermenting, which contains E. coli, Enterobacter, and Klebsiella. While the fecal coliforms, they are, means obviously because of the fecal contamination, these coliforms come into act, and these are also aerobic facultative anaerobes. And gram-negative organism have non-spore forming rod shape, but they ferment the lactose at 44 degrees Celsius. That's the peculiarity of fecal coliform, and you can differentiate fecal coli from coliforms with this temperature thing, like 44 degrees Celsius, right? When we talk about thermodiuric, temperature, diuric can sustain, like the organisms which can survive the pasteurization condition but cannot grow, those are known as thermodiuric bacteria, which contains the Mycobacterium lacteum, Bacillus cereus, Bacillus licheniformis, Clostridium, and Alkaligens and Streptococcus faecalis. Thermodiuric. Now we will be talking about thermophilic. Thermophilic, these are the organisms which can survive and grow at pasteurization temperature, which is a major concern for us, because when we say that the milk is pasteurized, we consider that milk is free of any microorganism. But these are the organisms which may play with your confidence of quality of that milk, like thermophilic bacteria. And this contains the Bacillus stereothermophilus, Bacillus circulans, and Bacillus thermoacidoreans, Lactobacillus thermophilus. And a major player which is also known as indicator organism, which is Coxiella burnetii. Coxiella burnetii is an indicator organism of pasteurization. It is called as indicator organism. Psychrophilic. Psychro means the philic means which love psychrophilic temperature. You know, which these are the temperatures. Psychro is the refrigeration temperature. they are able to grow at refrigeration temperature. Whenever we think that when we brought milk, and if you are keeping that milk in refrigeration temperature or in a fridge, you consider that no, the milk is safe because we have kept it at refrigeration temperature, and most of the growth of most of the microorganisms get retarded at refrigeration temperature. But these are the players who can spoil your game, like Pseudomonas, Acinetobacter, Flavobacter, and Archobacter and all. Then psychrotropic bacteria. Psychrotropic bacteria, their optimum temperature will be like below 20 degrees Celsius. See, here we are talking about psychrophilic, we are talking about 2 to 7 degrees Celsius, and psychrotropic, it will be below 20 degrees Celsius. There are very few which are classic psychrotrophs, and there are majority are mesophilic in nature, like dwelling between 20 to 32 degrees Celsius. Pseudomonas, Acinetobacter, Archobacter, and Bacillus coagulans. Now, the worst thing we will be talking about, the organism which should not appear in any milk will be the pathogenic microorganisms. In raw milk, obviously, most of these organisms get destroyed at pasteurization temperature, which consists of a group of Brucella abortus, militensis, Mycobacterium tuberculosis, Streptococcus pyogenes, Staph, E. coli, LM, then Leptospira, Bacillus anthracis, Clostridium, and Salmonella species. Again, the pathogenic microorganism in milk products are the challenges we are facing in our country, that the kind of product determines the kind of organism will appear in that particular, or it is vulnerable to certain type of microorganism, because we are having a variety of dairy products, which can be a fermented product, like which can be a coagulated, which can be a separated product, concentrated product, condensed, dried, frozen. We have variety of things, right? So, and the contamination can occur at pre-processing, processing, or at post-processing contamination level. Now, talking about yeast coming in milk, these are the aerobic and facultative anaerobes. They mostly grow at low temperature, responsible for slime formation. When someone talks about slime formation, then the first thing should come in your mind is about yeast, right? It imparts sweetish odor or surface, and it can impart the deep discoloration to the milk, right? Which is having array of species like Saccharomyces cerevisiae, then Debromyces candida, and Candida Saccharomyces toruplacus. So many are there. Then the thing which you don't want to see on your any sort of food will be molds, like aerobic and facultative anaerobes. They are filamentous, they are the spoilage organism, and they mostly grow at low temperature as well as at room temperature. So they are responsible for fuzzy or whisker like growth. See, this is the word you should keep in mind when someone talks about molds, this fuzzy or whisker like growth, and slime formation will be there, discoloration will be there. The similar thing which we saw in yeast also, there are also certain species of yeast which are responsible for slime formation and discoloration of milk and milk products. These mold species like Geotrichum, Candidum, Penicillium, Rhizopus stoloniformis, Mucor, and Aspergillus niger. Viruses, definitely there are so many viruses which can appear, mainly like cowpox virus, poliomyelitis, then Central European tick-borne fever virus, hepatitis virus, and the major concern of our industry will be like bacteriophages of lactic acid bacteria. Let's talk about the bacteriophages. You know, bacteriophages spoils your starter cultures. Starter cultures are basically used for the acid imparting, like preparation of dahi, and the fermented products, like develop for the different species of starter cultures imparts different flavor and texture to the product which is prepared, which also include dahi. And you can see the bacteria which are used in starter culture can be in a combination, can be in a mixed form, and can be the use with a multiple strain, right? So development of acid, and these particular starter cultures, as lactobacilli are growing in that particular product, it increases. When you are converting milk to dahi, definitely you are increasing the shelf life of that particular milk or the milk product. So it protects against the pathogens as well. Then definitely, then when we talk about the bacteriophages in milk, here you can see the Hollywood. You might have seen this kind of structure in Hollywood movies in 90s and decade. These are the viruses of bacteria. When we talk about bacteriophages, they are viruses of bacteria. Means what? They attack bacteria. These viruses of bacteria, they are non-self-replicating. They don't have their own mechanism of replication. They can't increase in number by their own mechanism. They have to take help of some other mechanism, or they have to hijack the reproduction mechanism of bacteria for their multiplication. So, but the most important thing, or the most important aspect of these particular bacteriophages is they are mostly host specific. Means what? If a certain bacteriophage is specific to certain organisms, suppose E. coli, it won't work against other species of microorganism. So you can, while getting rid of these bacteriophages or a problem of bacteriophage, we can take help of this mechanism or this phenomenon of these organisms, like their host specificity. Right? Again, in examination, it may be asked about the bacteriophages, how they multiply. There are basically two types of phages, like lytic cycle and lysogenic cycle. Lytic cycle will be let's see it in a diagram form. See, in lytic development, here you can see the organism, like that particular bacteriophage is sitting on that particular bacteria, which is also called as adsorption or infection. This particular bacteriophage will inject its DNA inside. Then that DNA is getting transcribed, and multiple copies of that particular DNA of bacteriophage is caused here. And after that, in late infection, the transcription leads to formation of different parts of that bacteriophage in the form of head, tail, and fibers, which again get assembled ahead, in phase assembly, next step. And then lysis of that bacteria occurs, that bacteria dies, and the newly born bacteriophages will come out, and that particular bacteria will die. Like a way, when these bacteriophages attack on your starter culture, it uses that particular organism, bacteria, for its reproduction, and that bacteria will die in this process, and that starter culture will not work, and your dahi will not be set. But when we compare with the other lysogenic method, in this method, lytic and lysogenic, right? Lysogenic, here what happens in this particular phenomenon, the organism, the adsorption will be there. After adsorption, DNA is inserted inside, right? That DNA in lytic cycle, individually it gets transcribed. Here it enters in host chromosome, bacterial host chromosome. It combines with host bacterial DNA, and then induction event occurs, right? Integration of lambda DNA replicates along with host chromosome. Here you can see. Then again ahead, after the induction event, again the synthesis of viral protein needed for formation of new virus will be there. Means these are the basic blocks, the protein blocks for the formation of new bacteriophages by using this prophase pathway. Again, rapid replication will be there of that DNA packaging, and then cell lysis releases a large number of new viruses. Again, the only thing you should keep in mind while differentiating lytic and lysogenic, in lysogenic cycle, the DNA of bacteriophage will get integrated with chromosomal DNA, chromosomal DNA of that bacteria. That's the only difference. Then how you can prevent the bacteriophages? So there are certain ways of preventing that bacteriophages, as these bacteriophages, they are specific to a certain species of bacteria. You can go for rotation of starters. You can use another version, what do you say in Marathi, so rotation of starters. Then bacteriophage intensive mutants, you can genetically modify that particular organism, bacteriophage, or your culture itself. The organisms of starter culture, if they are modified, then it will be like it won't be able to grow the bacteriophages. Then the media in which you are growing that your starter culture, which should include phase inhibition media. There are certain you can integrate certain constituents which are causing the inhibition of phases. Then prevention of airborne contamination, you should handle that particular starter culture in a proper manner. Then cleaning and sanitation of utensil, preparation of aseptic cultures, genetic manipulation of starter cultures, and some other methods are also there. Genetic, like radiation and all that. Genetic manipulation of starter culture. Now let's talk about the food poisoning. When you talk about bacterial food poisoning, bacterial food poisoning हुआ है, मतलब क्या हुआ है? There are 3 ways of occurring bacterial food poisoning. One thing is infection, other is intoxication, and third one is toxico-infection. In competitive exam, most of the time, the differentially these 3 terms are asked. When you talk about infection, the bacterial food infection, it refers to foodborne illness caused by ingestion of food containing viable bacteria. Means you are eating that particular pathogenic bacteria. The infection, food infection is there, like Salmonella, Listeria, pathogenic E. coli. Then that organism will grow and multiply in the host, leading to the illness. When you talk about intoxication, intoxication will be like bacterial food intoxication will be, it is caused by the ingestion of food containing preformed bacterial toxin. Preformed मतलब फूड में पहले से भी टॉक्सिन्स मौजूद है. लाइक एग्जांपल्स है स्टेफिलोकोकस ऑरियस एंड क्लॉस्ट्रिडियम बोटुलिनम. देन these toxins, they will be causing the illness. When you talk about toxico-infection will be refers to when bacteria like Clostridium perfringens present in food are ingested that subsequently produce toxins in the host. मतलब क्या है? When you talk about infection, organism अंदर जा रहा है और infection कर रहा है. Intoxication में toxins अंदर जा रहे हैं फूड के साथ और illness कर रहे हैं. But when you talk about toxico-infection, at that time, the organism is entering in the body, producing toxin there itself and causing the illness, right? So when we talk about infection, their examples are like Salmonella, Listeria monocytogenes, E. coli, that too of enteric type, Shigella, Campylobacter, Yersinia, Vibrio, and Aeromonas. When you talk about toxico-infection, it will be like Bacillus cereus, again of diarrheal type. Then Clostridium perfringens, Vibrio cholerae, and E. coli, which is enterotoxigenic in nature. Intoxication will be, when you are talking about intoxication, it will be like Staph aureus and Bacillus cereus, which is of emetic type. Here Bacillus cereus of diarrheal type. Many times it is asked in the examination. Emetic type in intoxication and diarrheal type in toxico-infection. Then Clostridium botulinum. Clostridium botulinum, see, this is the favorite question of all competitive exam. Clostridium botulinum. Whenever someone is talking about Clostridium botulinum, he may ask in the question that when he is giving the word Clostridium botulinum, in answer he will be expecting canning, and when he is asking in canning in the question, maybe which is the indicator organism in case of canning, the answer will be Clostridium botulinum. So when these together, whenever someone is asking about botulinum, a word should come in your mind. Very first will be like the canning. So it is a list of the milk-borne infections and all those infections in detail. To save the time, we should not read all those things right now here. So salmonellosis, which is caused by typhi, paratyphi, and all those. Salmonella species like enteritidis and others. Then shigellosis, Shigella dysentery, laboratory culture is there. Streptococcus pyogenes causes streptococcosis. Then here we should talk about the staph poisoning, Staphylococcus aureus poisoning. In this particular thing, in exam, the type of toxins are asked. They are compared with other toxins. Staphylococcus poisoning contains enterotoxins A, B, C1, C2, C3, D, E, and F. This is the peculiarity of this particular kind of poisoning. A, B, C1, C2, C3, D, E, F. A to F, just C replicate हुआ है C1, C2, C3, A to F. Now, when you talk about Clostridium botulinum toxinin, there toxin you can see A to G. A, B, C1, C2, D, E, F, G. उसमें क्या था? A to F, पर C1, C2, C3 था. यहाँ पे टॉक्सिन कौन से? C1 and C2, but the number increase here like G, A, B, C, D, E, F, G. Then E. coli poisoning, again when someone is talking about E. coli poisoning caused by E. coli, the kind of toxins involved will be like heat labile toxin and heat stable toxins. Those both of them will be like enterotoxins. Fungal intoxication, Aspergillus flavus and Aspergillus parasiticus. Aflatoxinosis is a disease which is caused B1, B2. See, these are the things you can compare. We'll see. Aflatoxin B1, B2, B2A, M1, M2, G1, G2, and G2A. See, this is different. A is not here, right? C is not here. We are talking about fungal intoxication. B1 to 2A, B1, B2, B2A, M1, M2, G1, G2, and G2A. Then toxin infection will be Bacillus cereus poisoning. Bacillus cereus, as we discussed before, there are two types of poisoning: diarrheal type or vomitic type. Vibrio parahemolyticus, it causes the acute gastroenteritis. Clostridium perfringens, again enterotoxin. A, B, C, D, E. A and C are more pathogenic. A, B, C, D, E. It's not up to F even. A, B, C, D, E. No replication of C. In case of Clostridium perfringens. Clostridium botulinum में क्या था? अलग था. Now we'll talk about the meat hygiene. We are done with the milk thing. Now we'll very shortly we will try to go through the elements of meat inspection. The most important thing which we will which may be asked in the examination, like the cattle, the food animals. Let's talk about the food animals. The Bos taurus, European origin, will be there. Buffaloes, Bos indicus in Indian and African origin. Then Bos bibelis in buffaloes. In case of river and swamp buffaloes. Certain terms which are very important so far the exam is concerned, which you might have read in while learning the LPM course, will be like bull, which is uncastrated male bovine, heifers. These are the female up to her first calf. Cow will be a female having one or more cows. Steer or bullock will be the castrated male having castrated at 6 to 12 weeks of age. Stag will be a male bovine castrated late in life and therefore presenting more masculine conformation, stag, than the bullocks. Bullocks से ज़्यादा conformation अच्छा होता है body conformation stag का. Sheep and goat में the mufflin, Europe and Persia में mufflin species are there, domesticated sheep के. Then Uriel, Western Asia, Argali, the big horn. Then definitions like lamb, a sheep from birth to weaning time. Then tup or ram will be the uncastrated male sheep. Hog or wither will be the castrated male sheep up to at the age of 3 weeks to 3 months of age. Then Jima will be a female which is having not born a lamb. A female which has not born a lamb yet. Ewe will be a female having a lamb. Cast ewe will be ewe which has been removed from the breeding flock, right? Which will not be, cast ewe will not be used for further breeding, right? Sheep flesh will be mutton and goat flesh will be G1. That's the basic thing everybody knows. Then in case of pig, the pork pig will be like from middle white and Berkshire breeds of pig. Bacon pig will be from large white Essex, large black, Landrace, Welsh, Tamworth, kind of Tamworth breeds of the pigs. Ham pig will be the popliteal region of the buttock and thigh of pig. Again, the important definitions will be the boar will be the uncastrated male pig. Hog will be the castrated male up to the age of 6 to 8 weeks. Then stag will be a male pig with castrated late in life. Rig, a male pig with one undescended testicle. Rig is very important. A male pig with one undescended testicle. It is asked many times in the examination. Gilt or clean pig will be a female having which is not having any litter yet. Then sow will be a female having a litter. Then there are certain characteristics of meat. Beef, a young animal, light red color, tender in color. When you are taking a beef from an older animal, it will be darker coarse in with a dried appearance. Here, young animals having glossy appearance. In bullock meat, it will be varying light to dark red in color. Then, with a prominent marbling of fat. Marbling of fat is a concept. It is a uniform distribution of fat in muscle. That is called as marbling of fat. Then buffalo meat. Buffalo meat is also known as beef or cara beef. It resembles with beef, but it is more lean, tender, and having low level of cholesterol as compared to beef or beef obtained from the cattle. Fat of buffalo meat will be like crystal white in color and shows the very nice marbling of meat. Veal is a meat of calves which are exclusively fed on milk and slaughtered at the age of 2 to 4 months. They are slaughtered at the age when they will be weighing about 70 kg. Musculine will be poor and fat is white and jelly like. Barley beef, veal is a, see, these are the comparable concepts. Many times people get confused with these three concepts. Meat of calf having exclusively fed on milk and slaughtered at the age of 2 to 4 months. Barley beef, these are also calves after early weaning. They are fed exclusively on barley diet along with minerals and vitamins, and they are reared for 12 weeks, 2 to 4 months, 12 weeks, right? And then those are slaughtered at the age of having 90 kg weight at 70 kg in case of veal. Bobby calves, they are male calves, have dairy breed. They are slaughtered at the age of 1 to 7 days only. Bobby calf at the age of 1 to 7 days, because on dairy farm it is not affordable to rear the male calves. Then mutton refers to the meat of sheep. Everybody knows about it. Fat is white. Chivon, goat flesh or chivon resembles with mutton, but it is dark red in case of and it is coarse in texture. Fat is practically absent. Chivon shows typical goatee odor, particularly in males, which gets transferred from skin to carcass during the dressing time. Pork is the meat derived from pigs. Pork becomes nearly white on boiling. Horse meat is many a times used for the adulteration of the beef or the buffalo beef. Like the horse meat will be like dark red to bluish in color, which also turns rusty after some times of slaughter. Odor will be sweet and repulsive. In poultry, many a times the adulteration of meat of ducks, geese, turkey is also there with the chicken one. The broilers, broiler meat is properly known as chicken and whitish in color. Fat is deposited subcutely and it is tender meated with soft, pliable and smooth texture skin. When we talk about the spent hen or fowl, it is a meat from mature egg laying hen after their productive life. When the hen is done with its productive life or giving eggs, then that particular hen is slaughtered. Then it is called as spent hen. Then rooster is a young chicken, usually 3 to 5 months of age, of either sex, maybe male, maybe female, and it is tender meated with soft, pliable, smooth texture skin and maybe somewhat less flexible than that of broiler or fryer. Rabbit meat, it has a sweet smell, fat is firm and white. Deer meat, it is also called as venison. The flesh of deer is called venison. It is bright to dark red in color, low in cholesterol, fat is not marbled with meat. See, this is the particular thing which is differentiating it with the beef or the cara beef. Fat is not marbled with meat but has distinctive flavor. Now, certain points which are important regarding abattoir or slaughter thing. The water requirement for the various species at slaughterhouse will be like 454 liter will be required, liter of water will be required per pig, 454 liter per pig, 272 liter per bovine, 45 liter per sheep and goat, plus 25% extra for cleaning of slaughterhouse. These are the I have kept those slides in presentation which are important through your competitive exam. Like ventilation, it should be adequate. Talking about light in slaughterhouse or abattoir, the meat inspection should be preferably carried out in daylight. Adequate natural or artificial lighting should be there. So overall intensity of light, this is again area of question many a times. Like at all inspection points, the light intensity should be 540 lux. 540 lux. At workrooms, it can be like 220 lux. And in other areas, it can be like 110 lux. 1 lux is equal to 100 watts. All inspection points 540. Workroom 220. 540, 220. Then definitely at such a places there should be a continuous electricity with industrial 3-phase electricity is required at slaughterhouse, and standby generator should also be there. Then let's talk about some special conditions which are asked in examination about the slaughter. Delayed slaughter. What is delayed slaughter? Delayed slaughter, casualty slaughter, and emergency slaughter. We will learn these 3 terms comparatively. In delayed slaughter, there will be the animal is a fatigued, excited, and inadequately rested animal, and animals affected with transit sickness. Those animals will fall in this category. These animals will require rest, means along with the flock, those animals will not be slaughtered just because they are fatigued, excited, or inadequately rested. So we will keep those animals in larage itself and we will slaughter it in delayed manner, delayed slaughter. When you talk about casualty slaughter, definitely something went wrong with the animal. Then the casualty slaughter will be in case of where an animal is not in acute pain. You need not slaughter it immediately. It's not in acute pain, but is affected with a chronic condition. Chronic conditions like obturator paralysis thing, postpartum paraplegia, followed by milk fever. In such a cases, this casualty slaughter is adopted. Then the emergency slaughter. Here we will talking about acute pain. When an animal is in acute pain and suffering from a condition like fracture of limb, if animal get fractured in case of during transport and all, then this kind of slaughter is carried out, emergency slaughter. Let's revise this thing. Delayed, the slaughter is delayed in case of delayed slaughter. Casualty slaughter will be like it's not in acute pain, but it is required to be slaughtered at priority because of the conditions like obturator paralysis or the paraplegia. And when you talk about emergency slaughter, the animal is in acute pain, emergency acute pain like fracture of limb. Again, taking a review of stunning thing. Stunning will be a humane method of slaughter to reduce the pain of animal in a through animal welfare point of view. Animal is made unconscious and insensitive to pain before you are subjecting that particular animal for the bleeding, right? That is stunning. In many countries, the stunning is made mandatory. There are certain methods of stunning. Let's have a review of that thing. Puntila or awareness zone method. Second, percussion method, captive bolt pistol method, carbon dioxide gas stunning, and electrical stunning. Puntila method will be like, here in this picture you can see the animal is puntila or such kind of knife is used to awareness on the animal. Here you can see. In percussion, you can see the hammer here. The person is using hammer to make that particular animal unconscious. Penetrating type of captive bolt pistol. Penetrating, it's like a pistol you are using. It is kept at a certain point which varies. That point varies species to species. Like in sheep goat, it will be different as compared to cattle, buffalo, and even in case of calves. So, but the captive bolt pistol, you will be keeping that thing in on forehead of animal, the pistol, and you are hitting that particular animal with that pistol. But the disadvantage of this particular type of stunning is the very edible part. That brain is a very nice edible part of that meat obtained from that food animal. That part, the brain part, will get damaged with this captive bolt pistol, penetrating type. But in case of non-penetrating type of captive bolt pistol, the brain is kept, the brain part of that animal will be kept intact. Here only stunning will be there, hammering will be there. Then CO2 gas stunning. Again, this is also many a times they ask about the carbon dioxide percentage how much percent of carbon dioxide is used in case of gas stunning, and in which species this particular thing is used. So 70% carbon dioxide concentration is used in case of pigs only. This CO2 stunning is used for pigs. Bleeding of animals should be carried out within 30 seconds of stunning. Again, because the pigs, after this CO2 stunning, within 1.5 to 2 minutes, the pig will get conscious. So you have to do the stunning, slaughtering immediately after the stunning. There are 3 types, like oval tunnel, you can see in the picture. Pigs pass on the conveyor. Then deep lift. In this, it comprises of suitable size cage, which is lower the pig to a pit having CO2 concentration for certain time. Here you can see the pig is left in particular pit having CO2 concentration. Then Ferris wheel is there. It comprises horizontal revolving compartments containing the necessary concentration of carbon dioxide. Here, for the exam point of view, I am providing this particular thing. Recommended current and time characteristic for electrical stunning in case of pig, sheep, goat, poultry, and turkey in amperes and volts. Then slaughtering methods. Let's talk about slaughtering methods. In major methods of slaughter will be like halal, also called as Muhammadan or Muslim method. Second will be like jhatka or Sikh method. Third will be Jewish or kosher method. Then sticking, awareness zone. We have seen awareness zone thing, right? Pithing. These are the methods of slaughter. In that particular, the area of interest of competitive exam people will be like Jewish and kosher method. So we'll take a view of that thing. Kosher is the term applied to the procedure and techniques of slaughter. Kosher, applied for procedures and techniques of slaughter. That is known as kosher. You know, it's like a Bible. Kosher is a Bible of the slaughter thing, right? Term applied to the procedures and techniques. Everything is written about that kind of slaughter in kosher or that method. Okay. Then if it is done according to law of the religion, law of religion is called as Talmud. In Hebrew language, kosher means fit to be used as a food. Kosher means fit to be used as food. Then law of kosher. Then other regulations governing kosher slaughter are derived from Hebrew tradition referred to as Shehita. Just like you can remember this particular thing like Sanhita we used in our literature. It's like Shehita in case of other regulation governing kosher slaughter. Under these, animals are to be fully conscious, killed and bled thoroughly by clean stroke of knife, right? Then there are another term in case of kosher will be, and first term like 16-inch razor sharp steel knife. That knife is known as chalaf. CHALAF, chalaf will be the knife used, 16-inch knife used for the slaughter will be chalaf. It is stuck into the throat by trained slaughterer. The sohet, trained slaughterer, the person who is doing the slaughter is known as shohet, SHOHET, shohet. And then while performing kosher slaughter, kosher slaughter are animal is slaughtered without pause, puncture, pressure, tearing, and slanting. These are the terms. 5 principles, kosher's 5 principles. The slaughter should be carried out without a pause, puncture, pressure, tearing, and slanting. Then a specific organs of the viscera, lung, stomach, and blood vessel, they are examined by inspector, like a veterinary officer of their the duty performing like a veterinary officer at slaughterhouse is called as bodek. They used to call that inspector as a bodek, BODECK, bodek, right? Who will be seeking for the abnormalities, ruptures, and foreign matters, which we usually do in antemortem inspection. Then carcasses which are fit and those which are fit and passed by bodek are known as kosher, fit for consumption. Kosher, fit for consumption. And bodek will be the person, their veterinary officer, or the person who is carrying out the antemortem inspection is a bodek, like bodek. And if the condemned carcass is there, which is not suitable for eating, will be like trefa. See, let's revise this thing. Terms. Chalaf will be the knife. Shohet will be the person who is doing slaughter. Shohet, slaughterer, Bodek, veterinary officer, right? Kosher, fit to be used, right? And Shahita will be just like our Sanhita. Shahita will be in Hebrew tradition, the regulations are there regarding this slaughter, right? I hope everybody can remember these things. Many times these terms may confuse you in the examination. Let's talk about meat speciation. The meat speciation will be conventional methods, include some physical, anatomical, histological, chemical, biochemical, chromogenic method, immunological method, right? And molecular technique. So amongst those, the type of meat, this is anatomically you can see, this is a comparative chart. You can refer it afterwards once you receive this PowerPoint presentation. So you can read out and you can compare based on their color, consistency, and fat color, fat consistency. You can compare the kind of meat you have obtained or received for the meat speciation. Then many times they ask for the chemical method. Based on their chemical properties, you are trying to differentiate the milk, right? Trying to differentiate the meat based on the carotene content, iodine value, linoleic acid content, glycogen content, triacyl glycerol content, refractive index of that meat, or volatile organic compounds. So different fats are having different refractive index. In case of horse fat, it will be like very much like 53.5 refractive index. Cattle will be less than 40. Refractive index of cattle fat will be less than 40. Pig fat will be less than 50. Iodine number, horse fat will be very higher, 70 to 85. Lard means the meat obtained from pig, lard will be 50 to 70. Ox and sheep fat will be 35 to 46. Carotene content, in buffalo fat, it will be thoroughly absent, while in cow fat, it will be present. Fatty acid, horse fat, linoleic acid will be 1 to 2% in case of horse fat, and other fats, it will be like very minute, like less than 0.1%. In case of horse fat, it is 1 to 2%. Based on these chemical properties, you can differentiate the meat. And apart from that, there are so many new recently used molecular methods like PCR, nested PCR, and all those can be used for the differentiation. Let's take a review of meat spoilage. Let's talk about meat spoilage. The meat spoilage, it is nothing but the breakdown of carbohydrates. Breakdown of carbohydrate proteins, which may lead to discoloration, which may lead to cause of off odor, like production of H2S, SO2, indols, ketol, and amines, changes in texture because of the sliminess, and the putrefaction, which is a very deleterious thing. Then the spoilage types. What sort of spoilage can be caused? The aerobic spoilage, which involves the organism which requires oxygen for their survival, like spoilage by bacteria, fungus, yeast. Then anaerobic, there are certain species of bacteria which are responsible for causing spoilage of meat, but they don't require oxygen. They are anaerobic in nature. So this is a list, and many times it is asked surface slime. When some surface slime is there, you can doubt for the contamination of Pseudomonas, alkaligens, or Micrococcus, Streptococcus, and Leuconostoc. When there is a putrefaction, again you can suspect for Pseudomonas or Proteus. If color of meat is changing, then again some other species are there. Some spots are there. Red spot, if surface discoloration is there, red spots are appearing on meat, then you can doubt for Serratia species. Blue spot, then Pseudomonas cincinnati. Then if yellow coloration, discoloration is there, then you can suspect for Micrococcus and Flavobacterium. And if greenish blue to brownish black is there, then you can go for suspect for Chromobacterium lividum. And actinomyces are the organisms which are responsible for imparting musty odor to the meat. Then if stickiness is there, all sort of fungus, they will be causing the stickiness. Whiskers, we talk about these whiskers in milk spoilage as well. Mucor and rhizopus, right? Black spot is caused by Cladosporium or Aspergillus niger. White spot, Spirotrachum, Geotrichum. Green patches are caused because of the species Penicillium expansa. Fat decomposition can be attributed to the Thymenidium. Then of odor and taste is a peculiarity of all anaerobic spoilage. It can be in the form of soaring because of the virtue of the acid and gas which are caused because of the Clostridia and coliforms. Putrefaction is because of the foul smell. Taint is a bone taint. It is this particular taint thing is the term is associated with the pork. Then methods of detection. There are certain methods of detecting the spoilage like chemical methods, mercaptans, H2S indols, ketals, amines, dye reduction tests are there, resazurin, methylene blue, TBPN and TBA. Some physicochemical methods are used for the detecting the spoilage of meat, which involve ERV, that is extract release volume, meat swelling capacity, water holding capacity. You in objective type question, they may ask you which kind of method is this which is used for the detection of spoilage, which can be, you can classify it in chemical method, physicochemical and sensory method like color, odor and consistency. Then another important thing, so far the radiation, so far the preservation of meat is concerned, like food irradiation thing. It is controlled application of energy for ionizing radiation. The sources for that particular thing can be machines, electromagnetic waves, radionuclide, radioactive substances like cobalt-60 and so many alpha, beta, gamma rays. Then this irradiation, many a times it is an area of interest for the competitive exam. They used to ask about the kind of irradiation used for the food preservation. They may ask for the radioration, radicidation or radapodization. A low dose radiation is called as radioration, which is 1 to 3 kGy. Medium dose radicidation is for 2.5 to 10 kGy, and a very high dose of radapodization of 10 to 50 kGy. Now another term, a very important term which we can't forget to discuss about is HACCP. HACCP is nothing but hazard analysis and critical control point. Means being a quality control officer, if you want to observe or monitor the system, but it's not possible for a single person to take a look of all the batches which are passing or getting converted from raw milk to final product, then that particular quality control person can identify the critical control points of that particular process, and those critical control points can be observed regularly for certain interval of time. That is hazard analysis critical control points. That is HACCP. So when they will be talking about HACCP, there are 7 principles of HACCP. First, conduct a hazard analysis. Second, determine the critical control points. Establishing these critical limits means you will be deciding how, at what extent you can say that things are getting spoiled. Critical limits, monitoring procedures, then establishing corrective action, then record keeping, establishing verification procedures. That is HACCP. When you will be talking about the food safety standards, then there are international level, at international level, they will be talking about the organization which are involved in food safety. We will have WHO, World Health Organization, FAO, OIE, World Trade Organization, then Codex Alimentarius, SPS Agreement, then IDF and ICMSF. At national level, they will be talking about PFA Act and Rule, then BIS, MMPO, IMAC, APEDA, EI and IFoS Act. So this is recent happening in last decade, which should be discussed and which can be area of interest for the examination. The person who is setting the question paper is FSSI, it's the Food Safety and Standards Authority of India. It's a, see, priorly what happened now, since the independence, there were several rules covering different kind of foods. The people, those who are involved in malpractice of food adulteration or malpractices, other malpractices, they will be taking the shade, or they will be trying to escape from any punishment by taking the advantage of the difference, because of they will be claiming that this particular food is not coming under this particular rule, and they will be escaping from that. So there was a need of covering all these rules and regulation under one umbrella, for the food safety. So that was created with a one nation, one food law, that is FSS, Food Safety and Standardization Act. So priorly, you can see these are the array of acts which were present in our country, covering different kind of food products. Prevention of food adulteration, fruit products in 1955, meat food product, vegetable, edible, oil packaging, different, almost more than 8 are there actually. Those were there, but those were not sufficient to catch the malpractices. So the FSSI has been established with for protecting and promoting the public health through regulation and supervision. In 2006, the Food Safety and Standardization Act was passed in Parliament in 2006, but the regulation, the rules were created in 2011. That's the again area of interest for the examiner. Food safety standard rules were made in 2011, right? The act was passed in 2006. The FSSI is basically authority. You can see the structure of FSSI. The food authority will be governed by the chairperson and 22 members, which are assisted with the food standards thing, which consists of scientific committee and scientific. So 17 scientific panels are there, which helps for the chairperson. Then for the compliance and inspection, see, to set the standards, they do have scientific committee and panels. For compliance and inspection, they are having central advisory committee, state level steering committee, and district level advisory committee. The FSSI, it works at 11 divisions, different 11 divisions are there through which it carries out its work. At the time of FSSI establishment, the first thing they carried out was they categorized all sort of food in main categories. They created almost 16 main categories of food, out of which first category is dairy products and analog of our interest. 8 is a meat and meat product, including poultry. 9th is fish and fish product, and 10th is egg and egg products. So in total, FSSI divided all sort of food in 16 categories, and a special category, 17th category, which covers the substances which are not for direct consumption as a food. So that's all about the food safety. So far, the veterinary public health subject is concerned, or the syllabus of your exam is concerned. We will be talking about the zoonosis, epidemiology, and environmental hygiene in upcoming lectures. Thank you. Thank you so much.